So, hello students, good morning. So, today we will just go through some terms related with immunology. So, for example, the first in the dictionary is ABO blood group system. So, we know there are two basic systems of blood grouping, that is Rh positive or negative and ABO blood group system. So, in ABO blood group system, we see that basically there are four types of blood groups. Either they will have antigen A or B or both A and B and no antigen. So people will have this blood group A, antigen A, they have blood group A, and people with antigen B have blood group B, and people with blood group this antigen A and B both have blood group this AB, and the people that have no A and B antigens, they will have blood group O. So the blood group system antigens are expressed on red blood cells. They are used for typing human blood for transfusion. Individuals who do not express A or B antigens on their red blood cells naturally form antibodies that interact with them. Then the next term is acquired immunodeficiency syndrome, AIDS. So AIDS is a disease caused by infection with the HIV-1, and AIDS occur when an infected patient has lost most of his or her T helper cells or CD4 T cells, so that infections with opportunistic pathogens occur. So basically these CD4 T cells are these Th lymphocytes or T helper lymphocytes. They defend our body, help our body to in defense against viruses and bacteria, against pathogens. So in AIDS there will be infection of HIV, which will destroy these T4 lymphocytes, and then our body will not be able to defend itself against opportunistic pathogens. So AIDS stand for acquired immunodeficiency syndrome, and HIV stands for human immunodeficiency virus. Then next they are active immunization. So immunization with antigen is called active immunization to distinguish it from the transfer of antibody to an unimmunized individual, which is called passive immunization. So there are two types of immunizations, active and passive. In some books they write active immunity and passive immunity. So active immunity is that in which this antigen or immunogen will be injected in the body through natural routes or through injection as a vaccine, and then it will activate this immune system and induce the immunity and will start production of antibodies itself. While in case of passive immunization, the pre-prepared antibodies, they will be injected in the body of a person. But the thing is that this active immunity is the permanent phenomena and this passive immunity is a temporary, because these antibodies, when they will be degraded maybe after 3 weeks or 4 weeks or 5 weeks, then the person will lose immunity. So passive immunity is temporary and active immunity is permanent phenomena. Then another term they are defining is acute lymphoblastic leukemia. Leukemia means cancer of white blood cells, cancer of blood. So acute lymphoblastic leukemia is a highly aggressive, undifferentiated form of lymphoid malignancy derived from a progenitor cells that is thought to be able to give rise to both T and B lineages of lymphoid cells. So most of these leukemias show partial differentiation towards the B cell lineage, whereas a minority show features of T cell. So acute lymphoblastic leukemia, basically it's a cancer of blood. Acute phase proteins. So acute phase proteins are found in the blood shortly after the onset of an infection. These proteins participate in early phases of host defenses against infection. An example is the menin-binding lectin. Acute phase proteins. When they are defining this acute phase response, the acute phase response is a change in the blood that occurs during early phases of an infection. It includes the production of acute phase proteins and also of cellular elements. Acute phase response is a change in the blood that occurs during early phases of infection. So, so it can be a good source for the diagnostics and diagnosis of the disease and to control it in the early phase. Adaptive immune response. So adaptive immune response or the acquired immunity or adaptive immunity is the response of antigen-specific lymphocytes to antigen, including the development of immunological memory. So adaptive immune responses are generated by clonal selection of lymphocytes, and adaptive immune responses are distinct from the innate and non-adaptive phases of immunity, which are not mediated by clonal selection of antigen-specific lymphocytes. So adaptive immune responses are also known as acquired immune responses. So the immune response which will take place in response to the exposure of our immune cells, that are lymphocytes, to the antigen, and then it will produce immune response and immunological memory, and it will involve this clonal selection of lymphocytes. So adaptor proteins. So adaptor proteins are key linkers between receptors and downstream members of signaling pathways. These proteins are functionally heterogeneous, but all use a similar domain known as SH2 domain as the means of interacting with the phosphotyrosine residues generated by the receptor-associated tyrosine kinases. The protein known as VAV is an adapter protein of this type. So, adenoids. Adenoids are mucosal associated lymphoid tissues located in the nasal cavity. So adenoids are the mucosal associated lymphoid tissues located in the nasal cavity. Adenosine deaminase deficiency. So the enzyme defect adenosine deaminase deficiency leads to the accumulation of toxic purine nucleosides and nucleotides, resulting in the death of most developing lymphocytes within the thymus. It is a cause of severe combined immunodeficiency. So it is a cause of severe combined immunodeficiency. So it involves this defect in the enzyme defects in adenosine deaminase. So adenosine deaminase deficiency can result in severe combined immunodeficiency because It will result in the death of most developing lymphocytes within the thymus. Then we come to this adaptive immunity. Adaptive immunity is immunity conferred on an eye or irradiated recipient by transfer of lymphoid cells from an actively immunized donor. This is called adaptive transfer or adaptive immunization. Then we come to this term affinity. So affinity is the strength of binding of one molecule to another at a single site, such as the binding of a monovalent Fab fragment of antibody to a monovalent antigen. So affinity is the strength of binding of one molecule to another. For example, the binding of this antibody to epitope on an antigen will be termed as affinity. So if it have more affinity, it means that antibody is better, and if it have less affinity, it means the antibody is not a good one. So that's why we see that in the primary immune response, the antibodies produced they have less affinity, but in the secondary immune response, the antibodies are better one and they have more affinity and they perform better. Then another term, that is affinity chromatography. Affinity chromatography is the purification of a substance by means of its affinity for another substance immobilized on a solid support. So in the chromatography we see that there will be column, and the column they will coat it with some, for example, they coat it with antigen molecules, and then if we have a mixture of antibodies and we pass that mixture through the column, the antibodies which will be specific for that specific antigen, they will bind there, and rest of these will be eluted. So we can use it for purification. So for example, an antigen can be purified by affinity chromatography on a column of beads to which a specific antibody molecules are covalently linked. Another term is affinity maturation. So affinity maturation refers to the increase in the affinity for the specific antigen of the antibodies produced during the course of a humoral immune response. It is particularly prominent in secondary and subsequent immunizations. Affinity maturation. Affinity maturation refers to the increase in affinity, that in the primary immune response the antibodies will have less affinity, but when this affinity maturation will take place in the secondary response, we will get better antibodies with more affinity. Then we come to the another term, agglutination. So agglutination is the clumping together of particles, usually by antibody molecules binding to antigen on the surface of adjacent particles. Such particles are said to agglutinate. When the particles are red blood cells, the phenomena is called hemagglutination. So agglutination is the process in which these soluble antibodies will help to link these antigens on the specific cells which are bound on the specific cells, or you can say the antibodies are free and antigens are surface bound, maybe on the bead particles or the red blood cells. So here they say that when the particles are red blood cells, the phenomena is called hemagglutination. So another term in this is allelic exclusion. So allelic exclusion refers to the fact that in a heterozygous individual, only one of the alternative C region or constant region alleles of the heavy or light chain is expressed in a single B cell, and in an immunoglobulin molecule. The term has come to be used more generally to describe the expression of a single receptor specificity in cells with the potential to express two or more receptors. Allele exclusion, we get two chromosomes, so it means two genes, so one gene have to be inhibited and one have to be expressed. So the first that will start expression, it will inhibit the other one. So if we have chromosome number 6 and chromosome number 6 is producing these antibody molecules, so one chromosome we get from mother and one from father. So if the first chromosome is producing this constant chain, heavy chain, then it will inhibit this heavy chain gene on the other chromosome. So allergen. So allergens are antigens that elicit hypersensitivity or allergic reactions. So allergens are the molecules which elicit type 1 hypersensitivity, and they help to initiate the allergy response. So if we want to get rid of allergies, it's better to stay away from allergens if possible. So allergic asthma is constriction of the bronchial tree due to an allergic reaction to inhaled antigen. So we have to figure out that what is the type of allergen to which we are allergic, and we have to stay away from that allergen.